Okay, so today we are going to finish the self-organizing map algorithm. We are going to finish talking about it and start a new topic, which is biological networks, construction and analysis of biological networks, which is the area that I'm working in actually. So last week, if you remember the self-organizing map, if you remember the components, we have a map, a 2D map, in which at each pixel of this map, we have associated vector, which has the same dimensions as our data objects. So we can have a pixel represent a data object by having copying the values onto that pixel. So each pixel has a position on the map, and in addition, at that location on the map, we can store a replica of the object, because at each pixel we have a vector associated with that pixel. Now, here's how if you remember, we have also went over a demonstration of self-organizing map. It was a Java applet, which was using colors as the objects to be clustered. So there are, and let's go over the algorithm once more. A self-organizing map algorithm has a fixed number of iterations. So it is user input. The user first says that I'm going to run the self-organizing map, this many iterations, 500 iterations, 1,000 iterations. So this loop here outside is that's the fixed number of iterations. For t is equal to from 0 to 1. That t, and how to increment that t, 0.1, 0.1, or 0.001, or 0.001. So how many iterations you are going to have depends on how much you are going to increase t. So increase t a small amount. How much this small amount is determines how many iterations you are going to have. So we first initialize the map, and usually a simple way to initialize the map is just by assigning random numbers to that map. So to that pixel positions, to those object vectors, we can assign random values. Non-existing data objects can be assigned randomly to the pixel positions on the map. Then in these 1,000, 1 million, or how many iterations the user wants, in these iterations, this is what we do. We randomly select the sample. This sample means that we select an object, data object, from real data, the data that we want to cluster, the data that we want to visualize, the real data. We select a random sample. If you remember the 400 students with 100 features example, so we select one student from those 400 students. Okay, this is the first step of the self-organizing map. And we look at the map and see which pixel matches best the selected sample. So this can be computed by this can be found out by computing the Euclidean distance, or any other distance you prefer, between the pixel values and your selected sample. So you compare your selected sample to each of the pixels, to each of the n-dimensional, 100-dimensional vector stored in that pixels. And initially, those pixel values were random. Okay, so at first, you have no idea where your sample is going to hit on that map. It can hit anywhere because they're all distributed randomly. It's going to hit someplace on the map randomly, meaning that the best matching pixel to the sample that is selected is going to be found by going over the map, and that pixel can be anywhere on this map. Then what we do is, that's the most important step. The third step here is the most important step of the self-organizing map. After selecting this pixel on the map, what we do is we update the values of the best pixel that we have hit on, we have matched, and we also update its neighbors. So previously, which was random, previously that pixel had some random values in it. What happens is that best matching pixel resembles now to our sample selected data point, and not only itself resembles the selected data point, this neighborhood up to a certain distance, up to a certain region around this best matching pixel also is updated to resemble the selected sample. So this updating the values is done in a way that if we are in the early iterations, when t is changing from 0 to 1, when t is close to 0, the update has more effect. I mean, the selected sample, for example, the best matching pixel at the first iteration completely copies all the values of the selected sample. It resembles fully, I mean, it's exactly a replica of the selected sample. But as the iterations progress, we do not completely update the best matching pixel. Instead, we use a weighted average of the current pixel on the map and the selected sample. So we use, for example, at iteration t is equal to 0.5, meaning that at midpoint in those iterations, we just simply take a direct average of the selected sample and the pixel on the map. We just directly average them. And as the iterations go higher, the selected sample is going to have a reduce effect on the map. So it's like the map is starting from higher changes happening on the map initially, and then these changes converges. It starts becoming like a tuning process. It starts reducing its effect towards the end of the iterations. Now, this is how we update the best matching pixel. So the other question is, how do we update the neighborhood? And which neighborhood pixels, how many neighbors? I mean, how many neighbors are we going to extend this update? And this figure shows, it shows a Gaussian peak here, and it shows how the neighborhood pixels are updated. The values, so if this is the x-y axis, the z values on this hill indicates the weight of the sample, selected sample, on those pixels. So what it shows is that the center of this peak is your selected pixel. It has that peak has a high value, meaning that your selected sample is going to affect that center more. And that effect is just a mathematical. What I mean by effect is just this, okay? Current pixel value times t plus sample value times 1 minus t. And since these current pixel value and sample value are vectors, we just, and this is a completely identified, so we multiply a vector with a scalar, we multiply another vector with another scalar, and we sum up these vectors. So you take an average of these vectors. So to give an example, if my selected sample is 1, 2, 3, and if the pixel on the map is, for example, 2, 3, 0, if this is the best matching pixel to this one, for example, and for example, I'm currently at t is equal to 0.8, okay? In the iterations, I'm at a later stage in the iterations from t is equal to 0 to 1. When t is close to 0, I'm going to, in the early iterations are indicated by t close to 0, and later iterations are indicated by t close to 1. So when t is equal to 0.8, what we do is, we do this. Current pixel value is this one, 2, 3, so this is the selected sample. Selected sample. This is the best matching pixel. So 230 times 0.8 plus 1, 2, 3 times 0.2. Okay, so if you do this operation, it's going to be what? This is 1.6 times, it's going to be 1.8, 2.4, 2.8, and 0.6. Okay? So the final pixel value, you update that current pixel value with these numbers. As you can see, the selected sample is 3, and the current pixel value was 0, but this 3 only could pull it, this number to 0.6. it could not pull itself to close to 3. It was 0 previously. It could only pull it to, if it was vice versa, if we were, for example, when t is equal to 0.2, if t was equal to 0.2, what would happen was we are going to have, let's also see that case, 2, 3, 0 times 0.2 plus 1, 2, 3, 0.8. It's going to be 0.4, 0.8, 1.2, 0.6, 2.2, and 2.4. Okay, if we were in early iterations, we would have 1.2, 2.2, 2.4, which is actually more similar to 1, 2, 3 compared to 2, 3, 0, and this one is closer to 2, 3, 0 in a later iteration. So if in a later iteration, the selected sample has a reduced effect on the map. Okay? Each of these selected sample changes the map values. At the end, the map is going to look like a clustering of our objects. But in the early iterations, we have more change. We update the map more drastically compared to later iterations. So this is the best matching pixel. So we do a similar thing. We do this. All we do for the neighboring pixels is that even we are in a even when we are in an early iteration, what happens is that if you look at this Gaussian here, you can think of the peak as t. Okay, the peak of this hill, the z coordinate of the peak of this hill can be considered as the value t. Okay, if that value is t, it means that actually, if it's t in the early iterations, so let's see. Is it 1 minus t or t? I'm trying to find it out. Yeah, it's like t. Okay, because when it's 0, it's going to be high. The sample value is going to affect more. So it's like similar to t. So the peak of these hill is you can consider as the iteration variable t, and these numbers, as you can see, the height of the hill decreases as you go from top to the neighbors. Okay, for example, look at this neighbor pixel here. That neighbor pixel here is like 10% of the t value. It means that the effect of the sample is even reduced at that point. Actually, it's the selected sample. If you reduce this, we are increasing the sample. Actually, it's like 1 minus t. I'm confused here. So what you need to know is that we use the same weighted average for the neighboring pixels as well. But the neighboring pixels, the effect of the selected sample on the neighboring pixels decrease even more compared to the current pixel. Even we are in an early iteration, the neighboring pixel are going to affect less compared to the best matching pixel. And this effect can be done by using a Gaussian function like this. By multiplying our t with this Gaussian function, we can, based on how close that pixel is to your current pixel, you can adjust the effect of the selected sample on that pixel. Okay, as you move away from the current pixel, the effect of the selected sample is going to decrease. And that decrease is again done by using a weighted average of the selected pixel and that current pixel at that neighboring position. Okay, so we have went over to how to find best matching pixel. We can use a distance like the Euclidean distance for that. And here update the pixel values the best matching pixel and its neighbors are updated like this. We can use a Gaussian function to determine how the neighbors are going to be affected. Yes?
So in order to understand the way I'm asking, we first calculate by this function
the weight, yes.
After that, we normalize by the Gaussian.
Yes, exactly. So now, the algorithm is this simple. I mean, only 4 steps. You continue doing this, and in this, the first step, randomly selecting a sample, it's selection by replacement, meaning that after you select that, after you use that sample, you put it back into your bag of samples. It means that in the second iteration, there's a chance that it's going to be selected again. So you may have 400 students, but you can run 1,000 iterations or 200 iterations. If you use 1,000 iterations, one student is going to be at least selected 3 times. At least some students need to be selected at least 3 times if you're using 1,000 iterations on 400 students. Okay? By using the pigeonhole principle again. I mean, with 400 students, 1,000 iterations, one of the students has to be selected at least 3 times in order to fill those 1,000 iterations. You can also have 200 iterations, 100 iterations, meaning that some samples may not be selected at all. This is all completely random. There may be some samples in your data set which are not touched at all by this algorithm. Okay? The first step, randomly select a sample, makes this a randomized algorithm. You do not guarantee to look at all your students. There may be some student in your data which you do not use the features of that student. And at the end, we end up with a map like this. And this map, which is the grayscale representation, is also called, I think, the error surface. This visualization of the map is called error surface, which shows how the pixels on the map at the end, how the pixels on the map are similar to each other. Initially, this map is just looks like a noise, okay? It's like a noisy TV monitor initially, and at the end, it becomes regulated, and the clusters appear like this. And you try to find out the islands or bumps on this map, which represent pixels with similar values. And those pixels should have a certain representation in data, in your real data. One thing, this is also really, you need to think about this. The pixel values here, if you select a pixel from your map, from your final map, there may not be an exact matching data point, and it may not correspond to a real student. Okay, this is what I want to say. I mean, at the end of your self-organizing map, if you just look at one pixel to find out where a student falls, or you may wonder this, for example, at the end. After you have this map, it gives you an idea about the general features of your students' general clusters. But if you wonder this, if you select a real student and want to find out which class this student belongs to, which category, which group this student belongs to, that student might not have an exact pixel on the map, because our maps here are just is a weighted average of many data points and the existing pixels on the map. At the end, if the values of a pixel, the high dimensional vector values of a pixel, may not correspond to an exact student exactly at the end. So how do you know, given student A, which group, what the question is, if I'm asking, given a student A, which of these groups that student belongs to? How do you find that? I mean, if these bumps here represent my groups of students, and your goal at the end is, given a student, which group this student belongs to, or whether you may ask, let me ask a different question. I give you 2 students, student A and student B. are these two students in the same group at the end after this is clustered? How do you find that out? Exactly. So it's you use one of the steps of the self-organizing, which is the finding the best matching pixel. So what you want is student A, which cluster does this student belongs to? You just look at all the pixels on your map and pick the pixel which has closest distance to your student, and the group, for example, your student may fall into, may be closer to a pixel in this region. And in that case, the student will belong to this group, okay? And you do the same thing for student B. If it also falls somewhere similar, close by, within this bump, it means that they are in the same group. Okay, so you can, I stated the final thing just to tell you that the final map we have, in terms of pixels, they may not correspond to real students, the real data objects, because on our map there may be the number of pixels on our map may not be the same as the number of data items you have initially. You may have a 500 by 500 map, but you may have only 1,000 students, so much more pixels than the actual number of students. And that's all I want to talk about self-organizing maps. If there are some unclear points, you can ask some questions, or you may also, I suggest you do further reading on the subject if you want to learn more about self-organizing maps. But in terms of algorithm, this should be sufficient for you to understand how the algorithm works. There are some variations of self-organizing map in which doesn't use rectangular pixels, but they use hexagonal pixels, which has a more natural neighborhood relationships. Because if you, I mentioned this before, if you have rectangular data, whether this is going to be, is this going to be your neighbor or not? I mean, you're just touching this pixel with just one point. You don't have a shared edge with this pixel here. Okay, you're just touching it one point. If it was hexagons, we would share an edge with all of our neighbors. Okay, if we had an hexagonal map, we would have this. And there are also, to remove some boundary effects, meaning that some pixels at the edges of our map for example, a certain region here, the distance to a region here may be high on the map, but actually they may be close to each other. I mean, the corner effects, to reduce the corner effect, there are spherical maps. I mean, there are three-dimensional spherical maps. There's another variation like that. But under the underlying algorithm, in essence, is the same. Okay? However you organize your map, whatever your map two-dimensional manifold is, at the end, what you have is you have pixels, you have data points. These data points update your map at a number of iterations. And at the end, what you want to end up is two-dimensional relationships between your high-dimensional objects. Okay. Any questions? Yes. In this example, our data objects were three-dimensional. They could have been 10-dimensional. These are not pixel coordinates. Okay? These are our data objects that we want to cluster. This, for example, this best matching pixel can be at, for example, 100, 250 pixels. This may be the pixel coordinates of it. The pixel coordinates of a point and its actual data are two different things. The best matching pixel may be at this xy on the map, and these may be the values x1, x2, x3. It could have been 10-dimensional, it could have been 100-dimensional. Okay, the data of the pixel and the location of the pixel on the map are two different things. On our student example, each pixel is 100-dimensional, but they are still on a two-dimensional map. We put these 100 dimensions on 2 dimensions. Yes. And the neighborhood. When we update the map, we update the current pixel and the neighboring pixels. Exactly. And the distance of the neighbor to the current pixel determines how it is updated, how much it is updated. The longer we go from the current best matching pixel, the less they are updated. No, the samples, you cannot, the samples are fixed. You cannot change a student. Your data, you just read it. Okay, you don't change the data that you want to cluster. The data is read only. Okay, you just read it and try to build this map. Yes.
When you say in the middle of iteration, you do not update the best matching pixels.
We update, but less.
The formula.
Uh-huh.
I think you mean the normalization comes after the iteration. You mean that when you say in the middle, we don't do.
No, even in the middle we update. I just wanted to say that as the iterations progress, the effect of the selected sample is reduced. Okay, it's always updated. It's always updated, but the update, the effect of the update is reduced. In the early iterations, it just, for example, in iteration 0, the selected sample completely copies itself onto the current pixel. The current pixel values are gone in the early iterations, in the first iteration. As the iterations progress, the current pixel value also is retained. We just take a weighted average. The effect of the selected sample is reduced on the map. Yes?
We perform dimension reduction while solving.
It can be done. There may be a hybrid average. The main reason of self-organizing, what I understand, so your question is, principal component analysis, for example. So it would be 2 different competing techniques. The goal of self-organizing maps is to reduce dimensionality, actually. So it is capable of working on high dimensions. But as you can see, at one part of the algorithm, we perform a distance comparison between high dimensional entities. And this distance, its meaning of this distance, the distance may not be very meaningful as the dimensions increase. So you can couple self-organizing maps with other techniques to reduce the dimensions into a lower dimensional space and then do the self-organizing map. It can be done, but in general, self-organizing map is capable of working on high dimensional data. If you can reduce the dimensions effectively to 2 dimensions directly, you don't even need self-organizing maps. You can just reduce your dimension to 2 dimensions and just plot your points on those 2 dimensions. You are going to see the relationships between them. So it may be, what your friend asked is that there are other techniques for dimensionality reduction. They are not clustering techniques. They try to look at the data and see whether the data can be represented with less number of dimensions. One example are these points. Imagine I have these 2-dimensional points. Okay? All of these points, what do they have? They have x, y coordinates. Okay, x, y coordinates, each of these points. So they are 2-dimensional points. But to represent this data set, do you really need 2 numbers for each data point? So here, in order to represent this data, meaning that in order to uniquely, so this is my data. Each point, my data here, my data set consists of points, and each point is 2-dimensional, as you can see. Each of them have an x and y coordinate. So when I mention an xy coordinate to you, you can go ahead and find that data in my data set. Now the question in dimensionality reduction is that, is it possible to represent the same data in a lower dimensional space? And for this data set, yes, it is possible. As you can see, there's the way that I put those points linear on a line means that if, for example, this was my x-axis, all of the points would have y is equal to 0 coordinate, right? If this was my x-axis, if I had chosen my coordinates like this instead of, so this was the original coordinate axes, and all of them has different xy values. Now, if this was my xy coordinate, all of the data points would have y is equal to 0 coordinate. Means that y coordinates contain no information for those points. Only the x coordinates would be enough to identify each point uniquely. So actually it is possible to reduce the dimensions from 2 to 1. So there are techniques like this. Given in a higher dimensional space, there are techniques to find unique coordinate axes, which is going to capture the variation in the data best. And principal component analysis is one such technique that you can use. So you can use PCA in conjunction with self-organizing map, or use them separately and compare the results. Both can be done. Any other questions? Okay. If not, I'm going to start the new topic, which is biological networks. Was it like this? So I'm going to skip the gene regulatory networks because we only have 2 hours left. I'm going to, so introduction to systems biology and gene regulatory networks, we are not going to cover this. They are actually, part of it is covered by these construction and analysis of protein networks. And I'm going to talk about these in the remaining 15 minutes, and in tomorrow's 2 hours, we are going to finish our discussion of biological networks. First, we are going to start what they are. All right, it's here. Okay, so up to this point in the course, we have just seen individual proteins, individual protein structures, and maybe looking at their expression levels simultaneously for thousands of them. We have seen microarray data, we have seen sequence data, we have seen protein structure data. We try to look at their relationships in terms of similarity by constructing phylogenetic trees, or by clustering them, by grouping them together in terms of certain similarity measures, either sequence similarity or expression behavior similarity. We were able to group them. Now, in reality, not these proteins, I mean, they're gene products, they do not function alone in the cell. Now, in the context of biological networks, we will be looking at these proteins in their actual working environment, and we are going to be looking at the cells from a higher level. And this area is therefore called systems biology, okay? Looking at the whole cell, looking at all the relationships between what the proteins are doing in the cell, and try to understand this complex system is called systems biology. And important part of systems biology is these biological networks, networks of proteins. And here the grouping of the proteins are not going to be based on just their similarity, based on how they communicate with each other in the cell, how they perform the functions in the cell. So it's the actual wiring diagram of the cell is going to be in question when constructing and analyzing these biological networks. The term construction here refers to finding, I mean, we are not constructing it from scratch. The cell, in the cell, the network is already constructed. There's an existing network of interactions between proteins and their compounds. And the term construction refers to identifying these existing interactions in the cell. So in the cell, there are several proteins working together or communicating with each other to perform more complex tasks. So construction refers to the fact that which proteins are interacting with each other, what is going on in the cell. So taking a picture of the cell and trying to identify which proteins are acting together, what is going on currently. And here, when I say biological networks, I should also mention that these networks are highly dynamic. There is no static wiring diagram as in an electrical circuit. If you look at the electrical device, for example, if you just wonder, imagine, I always give this analogy. For example, a DVD player. Okay, a DVD player is a complex device. It has a CD reader. There are certain chips, some MPEG decoders in it. So it's composed of certain logical circuits in it, and they're combined together. There's a certain data bus maybe, which transfers the data from the CD to some output port. So it's a complex device. A cell is like that. The only difference is that it's highly dynamic. In the cell, some of these logical circuits appear and disappear under certain conditions. So it is much, much more complex. And in a DVD player, I mean, you may have like 100, 200, maybe 1,000 components at most. In a cell, we may have tens of thousands of components wired together in that crowded, very, in a cellular, it's a very microscopic space. So it's a very high complex space. And our system and our goal is to try to understand what is going on in a cell. Try to get a picture of these relationships. So this is the area of systems biology. Now we are going to be, and here this very simplified picture shows you some of the interactions that we can see in a cell. So this is the cell membrane. The ellipsoid is our membrane here. And what happens is that this cell is in communication with its outside environment. In the outside environment, there may be some chemicals, there may be food, okay? When I say food, it's like it's not like raw food. Again, in terms of molecules, like glucose or, so there are some chemicals outside, so the cell should be able to communicate with the outside environment through proteins located on its membrane. So on this membrane, there are proteins, and there are membrane proteins which are in constant communication with the outside environment. They can interact with these chemicals, and we have like a system bus, which the cell in the membrane, when it senses something, okay, so I have a toxin here, what should I do? It cannot just run to the nucleus and say, do this. So it has to relay this message to the nucleus so that maybe new proteins are going to be synthesized. So what we have in that case is called a signaling pathway. This signal that comes from the outside is propagated into the nucleus by interaction between multiple in a cascade of interactions, in a linear sequence of interactions. That signal can be propagated inside the nucleus, and the cell can act accordingly. And all these things are, there's no intelligent thing that can, there's no CPU. The CPU is like, everything is chemical reactions, okay? Everything is physical chemical interactions. So when I say this senses some toxin, means that a chemical binds to that protein, and this binding changes the shape of the protein in a way that is going to allow another protein to bind to that location. All of these things are physical binding. They're all chemical reactions that are going on. But at the end, the system is, although these atomic decisions are made by simple physical interactions and conformation changes, at a higher level, the cell is really acting like an intelligent being. All these things are, it's like programmed. So it's really amazing. It's really complex. And actually, I just want to, maybe I can go over very quickly, or maybe I can talk about it. And in the slides that I skipped, there was an example about an E. coli gene regulatory mechanism. So what happens is that under certain environmental conditions, the E. coli, a single-cellular organism, can decide whether to produce a certain enzyme or not. This enzyme is used to break lactose, so the E. coli can metabolize energy from this, can metabolize this lactose and produce energy out of it. But if there's glucose in the environment, the E. coli prefers to use glucose, but at the absence of glucose and presence of lactose, it's going to use lactose to produce energy. And based on the environmental, based on this if conditions, if this is there and this is not there, the E. coli can decide whether to produce this enzyme or not. And all these things happen by the interactions of proteins with each other in harmony like this. So this is the signaling pathway, and we also have genetic networks, meaning that presence of certain proteins, special proteins known as transcription factors, interact with the DNA directly. They can physically bind to DNA to start a transcription of another protein, another messenger RNA, or not. So there are high level, there are really complex regulatory mechanisms that are going on in the cell. We call these regulation type of interactions a genetic regulatory network. And there are also metabolic pathways. Metabolic pathways are about having a single chemical compound, and by a successive sequence of reactions, this compound is turned to another compound, and some metabolic pathways are in cycles. They can, for example, the citric acid cycle, for example, there's known metabolic pathways. And here a compound is turned into different type of molecules in successive reactions. So, and there are hundreds of these different pathways in the cell, and it's very dynamic. So it's a very complex system. And what we mean by biological network construction is, for a given organism, for a given tissue, identifying these wiring diagram of that cell is our ultimate goal. And this is still a really hot current research topic. If we can find this blueprint of cells, we can understand how disease occurs. We can have ideas about how to cure diseases. We can have ideas about how certain biological things happen. Right now, for example, lots of drugs on the market, lots of medical drugs on the market, we don't know the actual process, how does it cure the disease. Some of them are not known. They're just by trial and error type of drugs. I mean, the actual disease mechanism, the biological, the disease process, most of them are not known. But if we have more knowledge about these pathways and interactions, we'll be able to design more targeted drugs, which we know the side effects before we create them. So this is really the ultimate goal of bioinformatics and systems biology. Now, right now there are low throughput studies in many molecular biologists, for example. They focus on a specific pathway, a signaling pathway, for example, or a specific metabolic pathway, and it may be finding all the details of a single metabolic pathway may be a PhD thesis. Okay, so it's really, I'm saying this not to decrease the value of a PhD thesis, but to say that this is really difficult. Okay, even working on a single pathway and finding out all the details of it is very difficult process. But luckily, in the last 20-30 years, many researchers have found some pathways, information about some pathways, and they are accumulated in databases. And as bioinformaticians, what we do is we collect all these low throughput pathways from different places and try to see the whole picture together by combining these individual pathways into what we call networks. The whole network determines how everything, because even these pathways, as you can see, pathways also may interact with themselves. The signaling pathway may initiate a metabolic pathway, so there may be inter-pathway communications, inter-process communications within a cell. Now here's, and these networks accumulated, this accumulated data stored in terms of networks are available publicly in many different databases on the web. And there are different types of networks. There are protein-protein interaction networks which focus on physical protein-protein bindings, protein-protein interactions. There are pathway databases like KEGG, which focuses on, for example, metabolic pathways or signaling pathways. And in signaling pathways, also the interactions are physical protein-protein interactions. So some of these databases, the information are overlapping. There are a lot of redundant overlapping information in these networks. So, and when I talk about a biological network, I'm going to be using this graph abstraction. Okay, we are going to be abstracting the interactions between proteins in terms of a graph discrete structure. We are going to have nodes, and we are going to have edges between these nodes. The nodes usually are going to denote proteins, and the edges between two nodes are going to indicate interactions between these two proteins. And this is a real network from the string database. String database contains literature created and predicted protein-protein associations. You can consider it as a functional association network, not a real physical interaction network, meaning that there's the difference between these two networks is how do you decide the existence of an edge? When do you put an edge between two nodes? Do you put an edge between two proteins when there is physical interaction in the cell, or whether they are part of the same pathway? There may not be physical interaction, but there may be members of the same pathway that act in different places. Again, you may decide to put an edge. In that case, you can say that they're functionally associated, and string is such a functional association network. And as you can see, we have many edges between, many nodes. And tomorrow, I'm going to continue this and talk about how we analyze these networks and how can we construct these networks by using some genomic data, by using sequence data, microarray data, for example. By using microarray data, can we construct such a network by using microarray data? Okay, so that's it for today. See you tomorrow.